Now we're going to change direction just a little bit and actually get to a mandatory part of the prokaryotic cell, one that's found in all prokaryotic cells, the cell wall. Cell walls are found on virtually all prokaryotic cells, but the cell wall structure is actually very, very similar regardless of the kind of bacterium. It's composed of a compound called peptidoglycan, or murine, and as you might be able to guess from its name, peptidoglycan, it has two parts. It has a carbohydrate, a sugar part, and that's what the glycan root in that word means, and a polypeptide or protein part, and that's what the peptido part. The sugars found in peptidoglycan are actually rather simple. You can see that these two chemical rings look very, very similar to the glucose ring found in simple sugars. Two sugars, N-acetylglucosamine, abbreviated NAG, and N-acetylmuramic acid, abbreviated NAM. And these two sugars, NAG and NAM, repeat a simple disaccharide over and over and over and over and over again. This repeated two-sugar polysaccharide gets cross-linked by the peptide part to form a very, very powerful and strong lattice structure. Here we can see this sort of illustrated in three-dimensional image. NAM repeats with NAG. This structure continues on and wraps all the way around the bacterium and comes back around and wraps all the way around the bacterium and comes back around and wraps all the way around the bacterium. And we can see these peptide cross-links holding the whole thing together. And these peptide cross-links make this peptidoglycan structure incredibly strong, very resistant to physical stresses. Now, bacterial cells show two different types of cell walls. We refer to these in class as gram-positive cell walls and gram-negative cell walls. Let's look at a gram-positive cell wall first. The gram-positive cell wall is made of peptidoglycan, but it's an extremely thick layer of peptidoglycan, that repeating NAM, NAG layer with peptide crosslinks is incredibly thick on the surface of this microorganism. We can see that there are some other things in there as well, teichoic acid and lipoteichoic acid, that add some further structure to this thick layer of peptidoglycan and also help the peptidoglycan to attach to the plasma membrane, the cellular membrane, which is down underneath here. What we find is that these teichoic acids also enable and regulate the ability of certain molecules to move from outside the cell to inside the cell. The main advantage of having this thick layer of peptidoglycan in the gram-positive bacteria is that an organism with this thick layer of peptidoglycan is extremely resistant to physical stresses, things that may try to affect that microorganism from the environment. The other kind of cell wall we find in bacteria is the gram-negative cell wall. It's also composed of peptidoglycan, which we can see right down here. But the peptidoglycan layer in the gram-negative cell wall is rather thin. Since the thin layer of peptidoglycan is not going to be as protective as a thick layer of peptidoglycan, the gram-negative bacterium is going to be somewhat more fragile than a gram-positive bacteria to physical stresses out in the environment. The physical shocks will be able to disrupt and kill gram-negative bacteria more easily than a gram-positive bacteria. But the gram-negative bacterium has another part to its cell wall. It's this outer membrane composed of phospholipids that is on the outside of the peptidoglycan layer. So where the gram-positive bacterium had a cellular membrane with a thick layer of peptidoglycan on the outside, two parts, the gram-negative bacterium has three parts. Here's the plasma membrane down here, the cellular membrane down here, a thin layer of peptidoglycan outside of that, and then this outer membrane outside of it. The big advantage that the gram-negative has because of this outer membrane is really that the outer membrane acts as a permeability barrier to things getting inside the microorganism, inside the cell. It acts as a barrier to things which are small and hydrophilic. Things like antibiotics, digestive enzymes have an incredibly difficult time getting from outside the cell out here to inside the cell. And what that means is that the gram-negative bacterium is going to be much better than the gram-positive bacteria at resisting the effects of antibiotics. This means that for a patient is that a patient with a gram-negative bacterial infection is going to have a much tougher time fighting off that infection with the use of antibiotics. Gram-negative bacteria are very important from a health standpoint for another reason. Here we're looking at a blown-up cartoon of the structure of the gram-negative cell wall. Here we have the cellular membrane, plasma membrane, with the cytoplasm inside the cell over here on the right, the thin layer of peptidoglycan here outside of the cellular membrane, and our outer membrane right here. Remember, you can have different components that are found in the cellular membrane. Have these globally circulates so that this fluid has very carbohydrate. Now, some lipid phosphorus, this one here, lipid A, both have extremely detrimental effects on the human body and really assist in the process of disease. Since these are found in all gram-negative bacteria, we now know at least one thing that's going to help any gram-negative bacteria, when it gets into the human body, begin to cause disease. Alright, now we're actually getting inside the cell. We're actually going to be getting to that part that separates inside from outside, the plasma membrane. Here we see a cartoon which illustrates the structure of the plasma membrane. It's composed of phospholipids, a lipid bilayer that acts as a permeability barrier. Many molecules can move from outside to inside the cell through the process of free diffusion, but the proteins that can move from outside to inside are going to have to be either small or hydrophobic, nonpolar molecules. If you're a large molecule, if you're a hydrophilic molecule, it's extremely difficult for you to move from outside to inside the cell unless you're helped along. We can see some of these processes, these passive processes, in this cartoon. Again, if you're a small nonpolar hydrophobic molecule, you can move from outside to inside from high to low concentration via the process of free diffusion. So a molecule that might be able to do this and move from out to inside from high to low concentration is a molecule like the oxygen molecule or the carbon dioxide molecule. Both oxygen and carbon dioxide are small nonpolar molecules, and if there's lots of oxygen out here and very little in here, the oxygen will freely diffuse right across that membrane. Let's also consider another rather small molecule, the water molecule, H2O. Unlike carbon dioxide, water is an extremely polar molecule, and consequently, water does not move from areas of high concentration to low concentration with any sort of frequency at all. It has an extremely difficult time moving across this membrane. Integral membrane proteins, or channels, facilitate the movement of water from outside to inside. The water moves from areas of high to low concentration. Through these channels, the protein helps the water to move from this area of high to low. Other molecules can enable the other different specific molecules to move from outside to inside from areas of high to low concentration. The process of diffusion, facilitated diffusion, and osmosis, which is a specific term of diffusion of water across the membrane, are passive processes. They don't require any energy to move, and in each case the molecules will move from an area of high to low concentration, and it's that chemical gradient that enables the molecule to move across the barrier. Here's a cartoon which kind of illustrates what's going on with this. Let's see what it says.
With their hydrophobic cores and charged surfaces, cell membranes are very efficient at blocking passage of many molecules. Some molecules, such as water, dissolved oxygen, and simple alcohols, are able to pass through cell membranes freely by passive diffusion. However, with their hydrophobic cores and charged surfaces, cell membranes are very efficient at blocking passage of many molecules. Some molecules, such as water, dissolved oxygen, and simple alcohols, are able to pass through cell membranes freely by passive diffusion. However, charged molecules and large molecules cannot pass directly through cell membranes, and hydrophobic molecules tend to become stuck inside the hydrophobic interior of cell membranes. In order to allow these complex molecules to enter and exit the cell, organisms rely on two forms of membrane transport: passive transport and active transport. Passive transport occurs along an electrochemical gradient. It does not require the expenditure of energy in the form of ATP for transport. Active transport requires the expenditure of ATP in order to transport molecules across the membrane. Active transport and some forms of passive transport require the use of transport proteins, such as the integral protein seen here.
Well, what happens when water moves in and out of the cell? As we've seen, water moves from areas of high concentration to low concentration. And because all cellular chemical reactions, or at least all the important ones, require the presence of water in order for the reaction to occur, a cell actually doesn't prohibit water moving from inside or outside under really any circumstances. Other charged molecules are tightly regulated. Consider the sodium ion or the potassium ion. Cells maintain very, very tight regulation of the amount of sodium and potassium that move in and out of cells. In large human cells, such as neurons, the concentration of sodium and potassium is critically regulated on the inside and the outside of those cells and play an important role in the process of transport, process of nerve transport. Here we can see what happens when a cell moves from an area of low salt to an area of high salt. If we start over here in panel A, we see that the concentration of salt on the outside of the cell and inside the cell is pretty much exactly the same. And what happens in this case is water moves freely back and forth in and out of the cell. If we take this cell and we move it to a new environment where the concentration of salt is very, very high outside of the cell, what happens is the sodium ions cannot easily move across the membrane, but the water does. And what happens is that the water begins to move outside of the cell, moves out from this cell, from in to out, so that the concentration of salt inside the cell becomes equal to the concentration of salt outside the cell. The result is that the cell begins to shrink and get smaller in size. If you happen to be a cell that has a cell wall, the same thing happens, but you don't see any apparent change in the size of the cell because the cell wall is a very rigid and thick structure. So a bacterial cell that we move into this hypertonic environment doesn't change in size at all, but it is going to be affected from its ability to grow because the loss of water from its cytoplasm is going to impair its ability to grow. Let's look at what happened when we move our hypothetical cell into an environment that has a very low level of salt in the environment. Salt, again, is not going to be able to move from inside to outside, but water is perfectly capable of moving. And what happens is that the water begins to move into the cell to make this concentration of salt in here the same as out here. And this cell will get larger and larger and larger in size until finally it begins to break apart in a process called hypotonic lysis. If we look at our hypothetical bacterial cell, though, cell surrounded by a thick layer of peptidoglycan, we can see that water is also flowing into this cell. However, the this cell is not going to get any larger in size. Consequently, we can see that this cell is going to be protected from hypotonic lysis because it has a very thick cell wall surrounding it. The movement of salt and the movement of water is very important in certain disease states in humans as well. The structure that's illustrated right here in this electron microscope picture is a cross-section of human intestine. We can see the interior, the lumen of the human intestinal wall, and the material, the tissue that surrounds it, the muscular tissue that surrounds and helps food to move through here. When humans ingest a microorganism called Vibrio cholerae, the causative agent of cholera, the bacterium produces a toxin, produces a protein that affects the cells that line the lumen of the small intestine. And what happens is that this Vibrio cholera toxin causes the cells that line the lumen of the intestine to begin to pump out sodium and chloride ions into the lumen of the intestine. The cells actually physically are going to lose salt because of the action of this toxin. This will create a hypertonic environment in the inside of the intestine relative to the cells that line the intestine. Well, what's going to happen when we're in a hypertonic environment? Water is also going to begin to move through the process of osmosis from these cells into the intestinal wall. So first, the toxin causes salt to accumulate in here, and then the water follows. What happens in cholera is actually a very alarming dehydration in the patient. The patient loses massive amounts of water, the volume of the stool gets larger and larger, and the patient has extremely violent diarrhea. Ultimately, the patient cannot drink enough water to be able to maintain hydration, and the patient will die of dehydration. Other processes that occur at the membrane are not passive. Active processes, active transport, require energy. Cells may expend energy to get molecules in when they are present in low concentration outside and in higher concentration inside. For instance, a molecule of glucose is very advantageous for a microorganism to take up, and a cell will use the process of active transport, requires energy, and bring molecules of glucose into the cell. So all active processes are going to require the expenditure of ATP energy, and will typically be used to move molecules in that the cell would like to have at higher concentration inside in comparison to outside. Let's look at just a couple of internal structures. The first of these are inclusion bodies. These are structures that are found in some microorganisms, and they're typically going to be used for storage. And you consider the process of active transport, where a microorganism would bring glucose into the prokaryotic cell because it's a molecule that a microorganism might want to store. Microorganisms will begin to accumulate these nutrients and pack them into certain much more energy efficient structures, energy efficient forms, which will allow it to keep more of this material inside than it would be able to if it just kept in free glucose. Inclusion bodies are generally pretty easy to spot under the microscope. They'll appear possibly as holes inside the cell, but they'll be filled with a very homogeneous mixture of certain kinds of chemicals. Endospores are structures that are also formed by certain kinds of bacteria, certain kinds of microorganisms, and these structures are produced by microorganisms to survive adverse conditions. Here we see a bacterial cell that's undergoing the process of binary fission and beginning to duplicate its DNA. But when certain environmental triggers are found and affect the cell, what happens is the cell doesn't divide into two, but the process of endospore formation is induced. As endospore formation progresses, one copy of the genome is destroyed in the process, And the second copy of the genome that was made during the DNA replication here gets surrounded by an extremely thick spore coat that surrounds to protect the endospore. Endospores are incredibly durable structures. We can see 3 steps in the process of endospore formation. Here is a new endospore beginning to form, and we can see the outer coat getting thicker and thicker and thicker surrounding it. It's incredibly powerful, very tight structure that protects that genome from extremely adverse environmental conditions. Here we see a field of bacteria. The bright green structures found in here are the endospores. We can see that some of the bacteria have endospores, others do not. If we were to look at this slide, look at this culture after another day or two, we would find that more and more of these cells have begun to form endospores. Well, what kind of environmental triggers cause endospores to be formed? They're typically going to be things where the microorganism is not going to be able to survive if it continues to grow as it had been in the past. Perhaps nutrients are running out, maybe the temperature's a little bit higher, perhaps the salt concentration has become very bad for survival of the organism. And the way this microorganism responds is to form an endospore. Again, some microorganisms can form endospores, others cannot. We can use the presence of endospores to help us to identify a microorganism. And I can't stress enough how incredibly durable this structure is. We've successfully been able to isolate endospores from ancient specimens, many hundreds or thousands of years old. The bacteria was last actively growing many centuries ago. And when we bring them into the laboratory, give them a good source of nutrition, we find that the organism then begins to grow very, very well again. So the survival of endospores is something that enables a microorganism to survive very, very poor conditions. Ribosomes are something that are found in every single prokaryotic cell. In fact, we find ribosomes in all cellular life, prokaryote and eukaryote cells. Ribosomes are composed of a bunch of components, small building blocks to make them up. Some of the components are made of nucleic acid, some of them are made of protein, and the whole thing assembles to form a mature ribosome. And what ribosomes do is they help the microorganism to carry out the process of protein synthesis, specifically in the process of translation. A messenger RNA will associate with ribosomes and transfer RNAs, and new proteins become synthesized with this. There's a number of antibiotics which are of current very good use. You may recognize some of these names like streptomycin or erythromycin. What these antibiotics do is they interfere with the ability of the ribosome to carry out the process of protein synthesis. You would think, since all cells, bacteria, and humans have ribosomes, that this might have very bad effects on the human body. But it turns out not to be the case, because prokaryotic and eukaryotic ribosomes, although they look very similar to one another and carry out much the same role in the cell with protein synthesis, there are actually some subtle differences between prokaryotic and eukaryotic ribosomes. And what this means is that the antibiotics that work to interfere with prokaryotic ribosomes don't tend to have a very big effect on eukaryotic ribosomes because of the differences in them. Archaea, we said, are structurally very similar to many bacteria. They resemble bacteria when we look at them under the microscope, but genetically they're incredibly different from a bacteria. We find that some of the things that we may see on the surface of archaeal cells look like things that we find on bacterial cells. But because of the difference in genetics between these two classes of organisms, although they serve the same function, they are structurally very, very different, chemically very, very different. One example is the humus that's found on the surface of many archaic cells. It looks like either a flagella or a fimbriae, there are long structures that help this guy to attach, and so functionally they're very, very much like fimbriae. But whereas fimbriae in bacterial cells just happen to be sticky, we can see that this hamus actually looks like a little tiny grappling hook. And so now you can see the physical basis of how this thing helps this archaic cell to actually attach. This hook structure actually grabs onto surfaces and holds this microorganism in place.